Good day, class. Batman, Spider-Woman, and myself, Mr. Moriarty, here to start with chapter 8.1 in your textbook, metabolism. So, yeah, we all want to have a higher metabolism because speaking in common vernacular, it means like, yeah, I want to lose weight or something like that. But in science, really, that's sometimes misunderstood. So what metabolism really is just the sum of all metabolic reactions, or the sum of all chemical reactions in your cell. So any kind of reaction occurring in your cell is metabolism. Now, reactions are going to require energy to be put into them at all times. Sometimes energy can be harvested out of them as well. So in that case, because it requires energy, yes, high metabolism will result in you losing weight and burning more of your fat reserves to use for energy. But metabolic reactions are regulated in response to your cell's need. So what metabolism really is, and we're going to talk about two major metabolic pathways in this chapter 8.2 and 8.3, is photosynthesis and cellular respiration. What metabolism really is, and what I want you guys to get out of this chapter is really think about how the body, how the cell, and in that case the body, regulates how things are done. So anything from how we build proteins and protein synthesis to when we need to grow more hair or make red blood cells or anything like that. All these things need to be controlled. And control has to be done in a chemical way. So it's really going to be about concentrations of things. So in our previous lecture, I talked about how when heat increases, things bounce around faster and we increase the chance of chemicals hitting into each other. Or if we add more substrate, there's more chance that it's going to hit an enzyme and we can increase the reaction. So all these ways are ways to regulate reactions. So as we go through this, in the back of your head, just kind of be thinking about how can the cell regulate processes. And as we learn more processes, think about how these are regulated and how really incredibly awesome it is that they are done. So here we go with metabolism. Make sure you're taking notes. I highly recommend by hand. You will have questions at the end, and you not only will have a test on this section, you are going to have a test at the end of the year that you need to know this stuff inside and out. So let's go. As I said, metabolism is the sum of all chemical reactions that occur within a living organism. So it's lots and lots of things. Like I said, the only ones you're really going to have to know are the metabolic processes in photosynthesis and cellular respiration. Well, I guess you will also have to know transcription, translation, and replication. We'll do that in our next chapter 7, which is before 8, but in our series after 8. Okay. Two types of metabolism that we're going to talk about. Anabolism. So I always like to think of anabolism as like anabolic steroids, which were really popular for like Arnold Schwarzenegger back in the day. But anabolic steroids, and when you think of steroids, people get really pumped up and big. So anabolism is a reaction where complex molecules are made from simpler ones. So I just think of anabolic molecules get put together, and then you have cannibalism, not cannibalism. That's when you eat people, like right back here. If I got hungry, like, tasty muscle. Oh, I just want to eat that muscle, but no, that's cannibalism. This is catabolism. And that's where things are going to be taken apart, releasing energy. I would take a few minutes. You also have these notes somewhere else to just go down through, like I like to make things into a chart. You'll see this a lot. This is actually from your textbook. So anabolic reactions, they build up complex molecules. There's something called endoergonic. So energy is put in this case, to the reaction. That's to put them together. Synthetic, meaning to build. So bio, life, to build. And examples, protein synthesis would be an anabolic reaction. Photosynthesis is an anabolic reaction. We're taking carbon dioxide and making sugar. Catabolism, our example is glycolysis. It's exergonic. Energy is taking out of the system. So kind of have an idea of those. More worried that you just know the basic of an anabolic and catabolic reaction. Okay, this is oftentimes one of the most missed questions when I do the test on this after 4 years. So just kind of, it's hard to take in and we get stuck with metabolism. But when I talk about a metabolic reaction, okay, so a metabolic reaction is going to be something that occurs in your cells that's going to require many intermediate steps. So maybe we start with our substrate here, or our substance A, okay? Substrate meaning it's going to be something that's acted on by an enzyme, and an enzyme is going to act on this substrate, and we're going to get a product here, okay? But this process is not done because we're going to have a new enzyme come in, and we're going to get a new product out of it, okay? And then we'll talk about this feedback thing coming up in a minute. But just know that a metabolic reaction, there shouldn't be an S there, sorry, requires a series of intermediate steps, and they're all going to be mediated by enzymes. They're going to be controlled by enzymes, and then they're also going to be controlled by something called end product inhibition, which we will come to later. Okay. These metabolic reactions typically can come in two ways. They can be cyclical, meaning the reaction kind of goes in a cycle. We're going to learn the Krebs cycle in depth. Maybe not this far in depth, but this is the Krebs cycle. We're going to start with acetyl coenzyme A, and it's going to work on citrate for our purposes. And then we have all these green ones are the intermediate enzymes. So even though you're not going to learn all these enzymes that go on with the Krebs cycle, know that it is a metabolic reaction and it's controlled by enzymes. So every little reaction in here has an enzyme doing its job, okay? And in this case, it's a cyclical one. It's going to go in a cycle. We're going to learn a lot about this coming up. But to make things happy for you, you only have to know citrate, oxacylate, succinate, and coenzyme A. Other than that, you don't have to remember all these other things, but you will have to know when this is reduced and oxidized. Oh, sounds so much fun. We're not going to worry about GTP at all, but we're going to talk about that in a minute. So just give you an idea, this is a cyclical big thing, metabolic reactions, enzyme mediated. Each step has an enzyme. Like I said, how does this control? What if I have plenty of ATP in my system? How can I shut this down? What's going to regulate that? Because we don't need this to just constantly be going on. If I have plenty, our body's good and it needs to slow down. So we'll talk about how that happens. So you could have cyclical, but you can also have it in series or a straight line. This one is actually not from human, but it's done in yeast, and it's the making of adenine. You guys know adenine. It's a base pair for DNA. It's actually pretty much exactly the same thing as ATP, which is adenosine triphosphate. Adenine and adenosine really, really close, similar pathways. But basically, if the yeast in this case doesn't have adenine, we're going to come back to this one later. If it doesn't have adenine, it has to be able to make it from other chemicals that it may have. And this is the process, each one intermediately done by an enzyme. So you can see it goes step by step by step by step by step. And even at one point, it actually has a choice to become ADP or GTP, and we'll say T because that's really what gets used. But really what I want you to see here is just that it can go in a series. Okay. To understand how this all works, we need to talk a little bit about enzyme inhibition. So to inhibit means to stop, and we're going to talk about two types of enzyme inhibition. We're going to talk about competitive inhibition and non-competitive inhibition, or allosteric inhibition. First one we're going to talk about is competitive inhibition, and basically inhibitors are going to be chemicals that reduce or prevent enzyme activity. Most of the time they're just going to reduce because they're not always constant. So the first type we're going to talk about is competitive, and what competitive means is we're going to have a chemical that competes for the same active site that the substrate would go into. So if I have something that fits in like this orange down here, it has the same shape as this chemical, and it's going to be able to bind into the active site of the enzyme. And with the induced fit model, this is allowed because it's not just lock and key, they're just similar enough. And if this is here, then the substrate can't bind to it, and therefore this enzyme, until it gets rid of this, cannot do its job, and we're going to reduce the ability of that enzyme to do its job. So substrate and inhibitor are very similar in shape, especially where the active site would be. That inhibitor is then going to bind to the active site, thus making the enzyme not be able to work. And it will be deactivated until the inhibitor leaves the site. And so what that gives you is a reaction kind of like this. Without the inhibitor, we're getting more enzymatic reaction. With the competitive inhibitor, much less. I mean, depending on how much competitive inhibitor, it could be much lower. Eventually, though, it kind of would balance out. If I have enough substrate concentration, you'd just be able to fill up some and not the others because of the randomness of bouncing into each other. I want to give you some examples of competitive inhibition that we use in medicine. One of the first ones is sulfa drugs, which are oftentimes called beta-lactam antibiotics. Okay, so this is penicillin. What it does is it binds to a active site in the process to make folic acid, and without folic acid, the bacteria will die. Actually, it's not able to produce its cell wall. And so penicillin just binds to the same active site as the substrate in that pathway to make the cell wall. And when it's bound, it can no longer make a cell wall. That bacteria can no longer reproduce, and we can pretty much get rid of the bacteria. So for those of you who plan on poisoning or killing people, cyanide is another example of competitive inhibitor. We're going to learn all about this beautiful electron transport chain coming up too, talk about a beautiful metabolic pathway. This is where ATP is created. And one of the enzymes that you don't have to know for this class, but maybe later, is cytochrome C, and it helps pass the electron along. Well, cyanide actually binds to the receptor in cytochrome C, and so when it's bound, we can't make ATP. Well, when you can't make ATP, your organism dies. That's not really good. So just some examples of competitive inhibitors right there. Know at least one example of a competitive inhibitor, at least be able to explain what it does. Oh, here's another one. I probably don't recommend this one on your IB exam, but Viagra actually is a competitive inhibitor. Nitric oxide is a natural chemical released in your cells. Nitric oxide causes vasodilation. So when you need to, you can actually get this to happen when you scuba dive too deep. It'll push nitric oxide out and cause you to do this. But vasodilation means your blood cells open up. So some scientists were trying to help with heart attacks by causing vasodilation, and they found that this drug, sildenafil, binds to one of the enzymes in the pathway that ends up shutting down the getting rid of your GTP, which is made from this, much more than you guys need to know, the nitric oxide. But what it ends up doing is it doesn't allow this part of the cycle to occur, which means we keep getting more of this cGMP, and that causes the vasodilation to stay, and increased blood flow to the penis causes erections. Those people are now very, very rich, even though they were trying to cure heart attacks. Brings us to our next one, which is non-competitive inhibition. We are going to talk about this one much more because it's used in our cells much more. And so where competitive binds to the active site, non-competitive is going to bind somewhere else on the enzyme. But when it binds to this enzyme somewhere else, it changes the shape of the active site. And if this active site has a changed shape, it's no longer going to work until our allosteric inhibitor or our non-competitive inhibitor is gone. Okay. So in this case, the substrate and inhibitor are not similar, and the inhibitor binds to the enzyme somewhere else besides the active site, and we call this an allosteric site. Okay, so it's another site on the enzyme where the allosteric inhibitor can bind to and thus shut down the enzyme temporarily until it opens back up. So it changes conformation, changes shape, and that's going to slow down the reaction rate. Here's how this one would look with substrate concentration going up and rate of reaction. No inhibition will plateau quicker. Non-competitive is going to slow down the reaction. And basically, this idea is how we're going to control reactions, because if I have a lot of something being made, well, then maybe the product that I'm making can actually inhibit one of the enzymes later. And as more products made, we get more competitive inhibition, which means we're going to get less product made. And as less products made, we get less competitive inhibition, and we get more product made. So you can see this balance where it goes up and down, and that's how the body regulates many of our metabolic functions. Okay, and this whole concept is called in-product inhibition. Big underlined letters there because this is super important. So the end product of a metabolic pathway attaches to the allosteric site on the first enzyme, therefore inhibiting or slowing down the reaction. So that's like, as you get more of it made, you have more opportunity for that final product to inhibit, which is then going to slow it down. But then once you have less, you make more product, and it goes back and forth, up and down. So when end product numbers decrease, the active sites are more open and the pathway becomes more active. And then when you get too much of the product, it slows it down until it needs more again. I'm going to come back to this adenine pathway. We're going to look at two different ones. They're just the ones I know really well. So in this one, you have all these are the substrates at each point and a new one, and really interesting. So for example, if you have a lot of AD2, it'll come back and it inhibits the enzyme air synthase. Okay, so if this is inhibited, we don't make any more. So if I have lots of adenine in my system, for example, it'll actually come in and inhibits, sorry, I meant to say that. Adenine actually comes up and inhibits this air synthase enzyme. So when we have lots of adenine, this pathway gets shut down right here. And won't continue on until adenine levels drop, and then it's more likely for this pathway to go through. There's also some other inhibitions that come here. But the whole idea is adenine comes and blocks this. Really cool in yeast, when adenine is not present, a lot of this gets built up and you end up getting this really pretty red color. Isoleucine, so unlike this, which is a nucleotide, isoleucine is one of our amino acids, and it is not an essential amino acid, which means we can make it from other amino acids. And so one of our amino acids that's required is threonine right here. And so this is the process that makes isoleucine. Well, when isoleucine's high, we don't want to put any energy into making more isoleucine because we have plenty of it in our body. So it comes back and attaches to the allosteric site on this threonine deaminase and inhibits it until numbers of isoleucine are low, and then we can turn it back on again. So if I'm eating plenty of this, we don't need to do this pathway. And that's how we regulate metabolic pathways. Never really going to test on this, but we've talked about how allosteric sites can cause inhibition. They can actually do the opposite and actually increase enzyme activity. This happens a lot in the making of protein. So in protein synthesis, you'll have allosteric sites on enzymes that'll make the enzyme actually make more protein or make more mRNA. Or in the process of the Krebs cycle, acetyl coenzyme A increases the reaction in the Krebs cycle. And so what happens in this case is when something binds to the allosteric site, it actually makes the enzyme work better. So just kind of know what that is. This is a possible test question for you. I'll let you guys kind of write that down, and then we'll end this lecture. Don't forget to answer your questions today, and have a wonderful day, and we'll have our next lecture next week. Here is the possible test question. Outline mechanisms of enzyme action. And I'll just let you guys read through those. I'd be excited to have one of those. Might even be a good idea to talk about end product inhibition, but that would probably be more for metabolic.